Hello and hi, my name is Muhammad Emanuddin bin Shahuddin. So today I would like to share with you guys about the physiology of our cardiovascular system, which is I will explain about a little bit about blood flow. Okay, let's have a look to this simple mind map. This simple mind map is explained to us about our blood flow in our body. So in our blood flow, it contains two types of blood, which is the oxygenated blood, which is bring the high concentration of oxygen, and we have deoxygenated blood, which is the blood that not contain any of oxygen. So this figure show our part of our heart, which is our heart contain of 4 chamber, which is left atrium, left ventricle, right atrium, and also right ventricle. Our heart also contain 4 types of valve, which is in our left heart contain 2 types of valve, which is aortic semilunar valve and also bicuspid valve. And on our right heart, there is also contain 2 types of valve, which is pulmonary semilunar valve and also tricuspid valve. These 4 types of valve is functioning to avoid the backward of blood flow. So our blood flow is start from the blood in our lung, which is the oxygenated blood from our lung, move into the left atrium through the pulmonary vein. This blood will continue flow into the left ventricle through the bicuspid valve. This oxygenated blood will be pumped out into our body through the aorta via aortic semilunar valve. Then the oxygenated blood will be changed into the deoxygenated blood because the oxygen in blood will be used in our body. The deoxygenated blood will be pumped back into our heart to the right atrium through the superior and inferior vena cava. So when this deoxygenated blood already entered into our right atrium, this blood will continue flow into our right ventricle through the tricuspid valve. And then when this deoxygenated blood already entered into our right ventricle, this blood will be pumped out into our lung through the pulmonary artery via pulmonary semilunar valve. So this blood flow is quite simple because we can memorize, which is our blood is starting from our lung, will be moved into our left atrium and then go to the left ventricle and they will be moved into our body. So the blood from our body will be get into our right atrium and go to the right ventricle and will be pumped out into our lungs back. And that's all from me, from Muhammad Emanuddin, about the physiology of cardiovascular system, about the blood flow in our body. I hope you guys enjoy to watch it. Bye bye and thank you.
Assalamualaikum and hi everyone. My name is Zuriani Hadirahana binti Alullah. I am from Environmental Health and Occupational Safety. My metric number is 2230078. I am honored to stand before you today to delve into the intricate world of the physiology of the cardiovascular system with a particular focus on the fascinating phenomenon known as the heartbeat. To begin our exploration, let's envision the cardiac cycle as a captivating dance performed by the heart from one heartbeat to the next. It is a rhythmic and essential process that keeps our circulatory system functioning seamlessly. Now, picture the heart as it squeezes and relaxes, akin to a well-choreographed dance routine. When the heart contracts, it enters the systole phase, and when it relaxes, it undergoes diastole. During systole, the heart undergoes a forceful contraction, expelling blood either into an adjacent chamber or into an arterial trunk. This contraction, for instance, propels oxygenated blood from the left ventricle into the aorta, ensuring the distribution of vital oxygen and nutrients throughout the body. Conversely, diastole is the phase where the heart relaxes, allowing the chamber to fill with blood. In the left ventricle, diastole facilitates the intake of oxygenated blood from the left atrium, preparing for the subsequent contraction. This squeeze and relax routine is not a one-time performance. It is a rhythmic cycle crucial for pumping blood throughout the body. This dance ensures that oxygen and nutrients are delivered to various organs and tissues while waste products are efficiently removed. In simpler terms, the cardiac cycle is comparable to a heartbeat dance, orchestrating the heart squeeze and relax movements to keep our blood circulating and our bodies thriving. Now, let's shift our focus to a critical measure of heart performance, which is the cardiac output. Cardiac output is a key metric that quantifies the amount of blood the heart pumps per minute. It reflects the heart's efficiency in circulating blood to supply oxygen and nutrients while removing waste. However, measuring cardiac output accurately poses challenges. The dynamic and intricate nature of blood circulation within the body makes precise cardiac output measurement challenging. One method involves the use of flow meters, devices installed in blood vessels to measure blood flow. However, this method is invasive because it is requiring surgical procedure. The FIG method and dilution techniques, on the other hand, offer alternative approach to measure cardiac output. The FIG method involves injecting a known quantity of a substance, for example a dye or gas, into the circulation. The substance then circulates in the blood and its concentration is measured during a specified period of time. By analyzing the dilution of the substance in the blood, a curve is obtained. The area under this curve can be calculated and related to the cardiac output. Essentially, it measures how quickly the substance is cleared from the blood, providing an indirect measure of cardiac output. Similar to the FIG method, dilution techniques involve injecting a substance into the bloodstream and analyzing its concentration over time. These methods, although more complex, offer crucial information about how well the heart is pumping blood. In summary, while cardiac output is vital for understanding heart function, its accurate measurement poses challenges. Invasive methods like flow meters are less favorable, prompting the use of indirect yet valuable approaches like the FIG method and dilution techniques. Now, let's turn our attention to the heartbeat itself, breaking down the intricate steps of the SA node activity and atrial activation to the ventricular contraction. The heartbeat is a symphony of orchestrated electrical and mechanical events unfolding in a series of precise steps. Here is a breakdown of each step. Step 1 is where the cardiac cycle commences, with electrical activity at the sinoatrial SA node, the heart's natural pacemaker. This activity triggers atrial contraction, initiating the heartbeat. Moving on to the step 2. The electrical impulse spreads across the atrial surfaces, stimulating contraction. It then reaches the atrioventricular node, allowing the signal to transition from the atria to the ventricles. Step 3 is where a brief delay at the AV node ensures ventricles are adequately filled before contraction. Subsequently, the electrical signal passes through the AV node, initiating both atrial and ventricular contractions. Then, the impulse travels swiftly down the interventricular septum, reaching the Purkinje fibers that distribute the signal to the ventricles and the papillary muscles. Lastly, Purkinje fibers ensure synchronized ventricular contraction, completing atrial contraction and initiating the forceful pumping of blood into the circulatory system. In conclusion, this intricate sequence of events orchestrates the heartbeat, ensuring precise coordination for the heart to efficiently pump blood and maintain overall circulatory health. Thank you for your attention, and I hope this journey into the physiology of the cardiovascular system has deepened your appreciation for the remarkable dance of the heart. Now, I would like to turn the floor over to the next person for the next topic.
What is electrocardiogram? Electrocardiogram is also known as ECG or EKG. At its core, ECG is a visual representation of the heart's electrical activity over time. It records the changes in electrical potential caused by cardiac muscle, depolarization and repolarization during each heartbeat. Electrocardiogram. Electro means electricity, cardio means heart, and gram stands for visualize. Depolarization is the process by which the electrical charge of a cell becomes more positive or less negative. In the heart, it specifically refers to the change in electrical potential that occurs when the cardiac muscle cells receive a signal to contract. Repolarization is the return of the cell's electrical charge to its resting state, becoming more negative again. In the context of the heart, it specifically refers to the recovery phase following the depolarization. ECG graph mainly consists of P-wave, QRS complex, and T-wave. The P-wave represents atrial depolarization. QRS complex signifies ventricular depolarization, and T-wave indicates ventricular repolarization. First is the P-wave. P-wave is the first deflection on the ECG. It represents atrial depolarization, the electrical activity that triggers the contraction of the atria. Typically lasts around 0.08 to 0.12 seconds. Next is PR interval. The PR interval is the time from the beginning of the P wave to the start of the QRS complex. It reflects the time taken for the electrical impulse to travel from the atria to the ventricles. Normal range for it is between 0.12 to 0.20 seconds. The QRS complex is a series of deflection following the P wave. It represents ventricular depolarization, the electrical activity responsible for ventricular contraction. Normally lasts less than 0.12 seconds. Meanwhile, ST segment is the flat isoelectric section following the QRS complex and preceding the T wave. It represents the early parts of ventricular repolarization. Normally, the baseline is at the same level as the PR segment. The T wave is the upward or downward deflection following the ST segment. It signifies ventricular repolarization, the recovery of the ventricles after contraction. The shape can vary and abnormalities may indicate cardiac issues. Lastly, the QT interval is the time from the start of the QRS complex to the end of the T wave. It represents the total time for ventricular depolarization and repolarization. Do you ever wonder how we calculate the heart rate? Actually, the heart rate is calculated by the RR interval.
The RR interval should be regular and consistent, especially if you are at rest, while it will shorten during exercise as the heart rate increases.
Do you ever wonder why ECG is important? ECG plays a crucial role in narrowing the coronary arteries, a heart attack, or an irregular heartbeat like atrial fibrillation.
Hi, I'm Wa'ai. I'm going to present about structure of heart for the left side. The left side of the heart consists of two chambers, left ventricle and left atrium, aortic valve and mitral valve. The left ventricle is the strongest and has an apex that points to the left. Let's continue with learning more detail for the structure of heart for the left side. So firstly, let's start with the pulmonary arteries. Left pulmonary arteries carry the oxygenated blood from left side of the heart up to the left lung. Left pulmonary artery also divided into smaller branches that reach the different segment of the left lung. Left lung contains alveoli where the blood exchanges carbon dioxide gases for oxygen. Meanwhile, left pulmonary vein carry oxygenated blood from the lungs to the left atrium of the heart. Another part of the heart is left atrium and left ventricle. Firstly, we're going to talk about left atrium that located on left posterior side. It acts as holding chamber for oxygenated blood from the lungs and then pump the blood that contains oxygen to left ventricle. As we can see on the diagram beside here, the left atrium has a thicker wall than the right atrium. This is because it exposed to higher pressure. Beside that, left atrium also has a small appendage called the left auricle. So, the location of left auricle is on the atrial surface of the left atrium. It's near the left pulmonary vein and the left circumflex artery. Left auricle contains pectinate muscle that help to prevent blood clotting. Next, let's discuss about left ventricle. So, maybe everyone will wonder how left ventricle of heart work. Okay, firstly, left ventricle is located at the lower chamber of the heart. Left ventricle receives blood from left atrium. Left ventricle also work together with right ventricle to forces blood out of the heart into arteries. Since the left ventricle forces blood to the other part of the body against a great flow of resistance, thus the wall of left ventricle is more thicker. Let's see inside of the left ventricle. As we can see on the diagram, The arrows show the mitral valve, or also known as bicuspid valve. Actually, bicuspid valve or mitral valve is found between the left atrium and left ventricle. Maybe everyone wonder how this valve formed. Actually, the answer is two cups from this valve, cups that attach to papillary muscle. Then this allow blood to enter the left ventricle from left atrium. Aortic valve is not located at left atrium. Actually, the location of aortic valve is between left ventricle and the aorta. Aortic valve open to allow blood from left ventricle to enter the aorta, and aortic valve close to prevent blood from flowing back into the left ventricle.
Pericardium is the membrane that surrounds and protects the heart. The pericardium consists of two main parts, which is fibrous pericardium and serous pericardium. Fibrous pericardium composed tough, inelastic, and dense, irregular connective tissue. It's open and is fused to the connective tissue of blood vessels, entering and leaving the heart. Fibrous pericardium also prevent overstretching of the heart, provide protection, and anchors the heart in mediastinum. Serous pericardium. The deeper serous pericardium is a thinner, more delicate membrane that forms a double layer around the heart. The outer parietal pericardium is fused to the fibrous pericardium. The inner visceral pericardium is called epicardium. Between the parietal and visceral layer of the serous pericardium is a thin film of lubricating serous fluid. It's also known as pericardial fluid. Pericardial fluid reduces friction between the layers of the serous pericardium. As the heart moves, the space that contains the few milliliters of pericardial fluid called pericardial cavity. Heart wall. First, endocardium. Endocardium is the deepest layer of the heart. Function of endocardium is smooth the lining to reduce friction to blood flow. Second, myocardium. Myocardium is the middle layer of the heart. The function of myocardium is the location of the muscle fibers responsible for pumping. Next, pericardium. Pericardium is the outer protective layer. It composed of visceral pericardium, pericardial cavity, and parental pericardium. Beside a triple layer sac that surround the heart, we have epicardium, pericardial cavity, trabecular cornea, coronary blood vessel, serous pericardium, fibrous pericardium, and heart wall. I'm going to explain about the right atrium and ventricle. The right atrium receives the oxygenated blood from superior vena cava, also receives the oxygenated blood from inferior vena cava, coronary sinus, anterior cardiac vein, and also the smallest cardiac vein. And all the structure will be taking the oxygenated blood into the right atrium. Once the right atrium receives the oxygenated blood from this vessel, will be pumping to the right ventricle through tricuspid valve. The wall of the right ventricle is thicker than the right atrium. The wall contains cardiac muscle fibers called trabecular cornea. The cups of tricuspid valve are connected to tendon-like cords. The cardiac tendinae are connected to cone-shaped trabecular cornea called papillary muscle. Blood passes from the right ventricle through pulmonary trunk to be oxygenated in the lungs.
Hi guys, my name is Hafiza Kimber Roslan. Now I want to discuss about our blood vessel. We just learned about the heart, which is the incredible organ that pumps blood all over your body to deliver oxygen to every last cell that need it. But what does the blood move through in order to get around? Blood travel through the blood vessel, and there are number of different types. So let's take a look of this now. First, we need to be aware that blood vessels come in three varieties. First, we have arteries, veins and capillaries. The first two, which are like the main highway, are easy to define. Arteries can carry blood away from the heart and they brush out as they go, becoming smaller and smaller. Veins carry blood into the heart, so they merge and become larger and they approach the heart. And then capillaries are the smaller vessels and make direct contact with the tissues around the body and supply them with oxygen, so these are like the surface street and alleyways. Now we have some terminology down. Let's learn a bit about the structure of blood vessel. This will be quite similar regardless of the type of vessel and most have three layers, which we call tunic that surround the lumen, which is the hollow part that blood flow through. The first is the innermost tunica intima, made of an endothelium that minimizes friction as blood travels, and most also have a subendothelial layer made of loose connective tissue. Next is the tunica media, made of smooth muscle cell and elastic fiber. This muscle is regulated by the autonomic nervous system, which can initiate vasoconstriction, where the muscle contract and the vessel narrow, or vasodilation as the muscle relax. This is important for regulating blood pressure and proper circulation. Lastly, there is the tunica externa, made of collagen fiber for protection and structural reinforcement. Within this layer, if looking at the large arteries and veins, there is a vasa vasorum, a system of much tinier vessels that supply the outer layer of blood vessel with nutrients. Now, let's get more specific regarding the type of vessel. The first one, we have arteries. Arteries come in the three groups. The first are elastic arteries. These are the largest and thickest, the one near the heart. These contain more elastin than the other vessels, so that they can absorb more pressure, which is necessary as they have to withstand the pumping nearby. Next, we have muscular arteries. These deliver blood to specific organs, and these are the most abundant. And lastly, we have arteriole. These are the smallest arteries, the one that lead into the capillary bed. Next, we move on to the vein. The structure of vein quite similar with arteries but have small differences, which is vein have thinner layer of small muscle and thicker layer of connective tissue. Predominant layer of vein is tunica adventitia. The wall of vein is thinner compared to the arteries. Vein divided into three categories. The first one is large vein. It have large diameter because it need to transfer blood into the heart that have high pressure. The second one is medium vein that carry blood to the large vein. Structure very similar to large vein. And lastly, we have veinule, smaller vein, carry blood from capillary to the medium vein. Endothelium as a dominant layer. Moving on to capillaries, these are the smallest blood vessels as we said. These have just a thin tunica intima, sometimes just one cell thick, often adorned with parasites, which are cells that stabilize the capillary wall. The lumen of the capillary is very small, just small enough for red blood cells to fit through in a single file. There are cutaneous capillaries, which are found in the skin and muscle, and these are the most common. Next, we have fenestrated capillaries, which are covered with pores along them to receive nutrients from digested food and to allow hormone to enter. And then there are sinusoid capillaries. These are found only in the liver, bone marrow, spleen, and adrenal medulla. So this is all you need to know about our blood vessel. So I hope you can understand and explain what is structure and function of our blood vessel. Okay, see ya.